Hi everybody, welcome back from lunch. It's great being here again. So I think I'm here for like the third or the fourth time, so I really love CGSI, all the interactions. And today I'm going to talk, I'm going to give a tutorial, which is, as you can see from the title, about predicting protein contacts or contact maps from sequence information. Now I'll just give a few words about background. So. I've been here, like I said, a few times, and it's a great meeting for all the genomics community, and there are people from PopGen or from cancer genetics and epigenetics, et cetera. But in computational biology, there is sort of a different community, which allows the structural biologists, right? And my impression is there is a disconnect between these two communities. Usually, if you go to Recomb or other conferences, usually they have their own problems and their own communities, etc. And there's not much interaction, I would say, between people doing genomics and people doing structural biology, which I think is either unfortunate or maybe an opportunity. So in the recent few years, there have been some synergy between using sequence information, in particular evolutionary information, to also help in problems in structural biology, and this is what I'll present today. And another disclaimer, this is not sort of the major research field that I'm working on. It's something we started to look at fairly recently, so I'll describe to you sort of an outsider's perspective on this field and my understanding of what is this field, what are the main problems, etc. All right. So, and of course, feel free to interrupt me with questions. So, let's give an outline. So I'll give a brief introduction to the problem of protein structure prediction, or computational structure prediction. And then I'll talk about how we can use sequence information, in particular multiple sequence alignments, to help us solve this problem. We'll talk about protein contact map, which is sort of one aspect of the protein structure, which we'll focus on today. And I'll present two methods which are based on graphical models. One's called Psycho, and another one called DCA, or direct coupling analysis. And then I'll describe more recent methods based on, of course, deep learning. So as you'll see, while the tutorial is focused on a biological problem, I'll try to give details about the statistical methods that are used in various stages. All right. So what is protein structure prediction? So it can be described very simply. So we have a sequence, right? So it's a string. I guess most of you are used to DNA strings or RNA, but this is the same. It's an amino acid strings. So the alphabet has 20 letters, representing 20 major amino acids. And the goal is to write a program or write a model that will take as input the sequence and will produce as output the structure. So it's this three-dimensional structure of the molecule. I see that some of this corner is cut, so whenever we have symbols there, I'll describe them. So we have a sequence S, right? So mathematically, it has length D. So you can represent it as an element of this A to the power of D, where A is a set of all amino acids, right, 20 amino acids. And how do you represent a structure? So we can use X, Y, Z coordinates, and they are in Rp. So P is different than dy, because D represents the number of amino acids, and P will represent the number of atoms in the structure. Each amino acid is a molecule composed of a few atoms. And to get the full structure, we need to, if you want to really nail down the structure, you really want to specify the physical location of each atom in the molecule, right? So P is some function of D, of the composition of the amino acids, but what we really care about are the positions of the amino acids and the atoms. And so we got this X, Y, Z, right? So we're really after a function f that takes as input the sequence and outputs a structure. Now, of course, there are some symmetries, right? You can rotate the protein or move it, etc., etc. But up to these trivial symmetries, this function, for the most part, is determined uniquely. Now, why is this interesting? I mean, this is, I guess, does not need any motivation. It's a classic problem in computational biology, perhaps the most classic, and there are quite a few Nobel Prizes given for experimental determination of protein structures, and even one for computational determination of protein structure. And you can see that the importance of the problem is not going away. If anything, it's becoming more important. So you can track down the number of known protein sequences. This is the blue on the left. As a function of time, and because next generation sequencing is so cheap and so ubiquitous, we see an exponential increase in the number of available protein sequences. Whereas if you look at the number of known structures, it is increasing, but at a smaller rate. So this gap between number of known sequences and the known structures is actually growing, right? So it's not keeping up with the genomic revolution. So what we would like to, right, so just to make it clear, the red bars represent experimentally known structures that are deposited in the PDB, that's the protein databank. So that's like one repository where people put the known structures. All right. So what is sort of the straightforward but quite difficult approach to determine structure? Well, a protein is a molecule, so it has to obey the laws of physics, right? So every molecule has energy, and like people after lunch, molecules too, they try to minimize their free energy, right? So there is this function, free energy, which is a function of both the locations of each atom, so this is R to the 3p, and also the identity of the molecules, right? So if on the same location we have different atoms or different molecules, the energy will be different. And conceptually, again, the problem is fairly straightforward. Once we write this energy function, delta E, then of course, we cannot change the molecule. The molecule is fixed, it's given, but we can change X, Y, and Z, the locations of the atoms. And we want to find X star, Y star, Z star of S. S is a molecule, which will minimize this function, right? So you can, in principle, write an optimization problem that is supposed to give you the structure. Well, of course, there are, right, so a protein is a chain, so there are all kinds of constraints because adjacent amino acids in the sequence, they also must be next to each other in the structure. But other than that, X, Y, Z are free to move and just to minimize this energy. It turns out that this energy function is quite complicated. And the way people in the field write it is it's a linear term. So we have some I of Wi EI. So, so molecule is large and complex, and you're trying to write the energy as a function of simple terms. So each EI depends on GI and CI, and GI is a local geometric variable, and CI is a local chemical variable, right? So for example, GI can be sort of if you have a chain, so you have the chain of the backbones of the proteins, you can have an angle between two amino acids. So this can be, for example, GI, and CI can be maybe a specific atom of one amino acid or a specific, and maybe a pair of atoms of adjacent amino acid. So the idea is to write many, many simple terms, each one involving only a few atoms or amino acids, such that the total energy will be the sum over all of them. So again, like I said, there are many, many, many terms, and if you just look at the most recent energy function of Rosetta, so Rosetta is one of the leading, if not the leading, programs for structure prediction. So these are all the different types of terms that go into this energy function, right? And in fact, there's a whole paper, this paper by Alford et al. That the title is something like describing the latest energy function of Rosetta, right? So it's really a complex function. And moreover, it's not really sort of fixed or known, right? So a common misconception, or at least a misconception that I had when I entered this field, was that, you know, because we have a molecule and we have the laws of physics, then the energy function is really known. And the problem is tough because it's a tough computational problem, right? So it's an exponential search space because the x, y, z coordinates of each atom can vary in the space. And if we have p different atoms, then the search space is exponential in p. But other than that, if we had infinite computing power, we could just maximize or minimize this energy, and we would solve the structure. Like I said, this is a misconception, and there is no agreement in the field about what is a correct energy function. And like I showed you before, the energy function is quite complex, and people are still working on changing and improving this energy function, right? So all this is sort of to motivate other approaches for protein structure prediction, which are, you know, maybe less based on physical principles and more based on data, right? A data-driven approach, and that's what we're going to do today. Now, again, if you want to think about this problem simplistically, you can think about the following data-driven approach. We have a bunch of known or solved structures. We have their sequences. So this gives us a training set, right? So we have these SIs and these known structures. We have a standard supervised learning problem. I agree. I mean, the labels are not simple. It's not a classification problem where we have 0 or 1 labels, or a regression problem where there is one continuous label. The labels are sort of complex. It's called a structured prediction problem. So the labels are vectors in R to the 3p. But other than that, it's a standard supervised learning problem. And you might want to build regressors just from the data for this problem. But again, even this approach is too naive to work because first, the function f we are looking for is very complex. And second, the number of labeled molecules, labeled proteins is, I mean, not small, but it's on the order of tens of thousands, which is probably not sufficient to learn this function. Yes, there was a question on the back, right?
I was just going to ask if you could stay behind the mic because it was really hard to hear you.
All right.
There was an air vent going. No worries.
So, okay, I'll stay here. So should I speak louder or it's an echo thing?
Either way.
All right.
A little bit louder.
Okay. Okay. So, again, we cannot just really tackle the problem as a standard supervised learning problem. And we need some, if you want, short routes or other approaches. So we look at two things that may help us. One is adding data, all right? So what do I mean by adding data? One can add unlabeled sequences, right? Like I said, there's a limited number of labeled sequences where we know the structure, but there's tons of protein sequences with unknown structure, which we can throw in. And this gives us a semi-supervised learning problem. So there are a few recent nice works about utilizing this approach, but that's not what I'm going to describe today. Another approach is for each sequence, you can add auxiliary information, additional information that may help you in prediction. Right, so we're still talking about the labeled data, the sequences where you know, in the training set you have your known structures, but in addition to the sequence S, we might add another information that can help us in prediction. A second thing that we're gonna change is, well, maybe predicting the entire three-dimensional structure is hard. So let's break it down into subproblems. And for example, one classical subproblem is predicting the secondary structure, so sort of the local structure of the protein, which is easier. And another, which we are focusing on today, is predicting a contact map, which is, from the name, I guess, even if you don't know this field, you can guess that what we're trying to predict is, for each pair of amino acids, do they touch each other, do they contact each other in the 3D structure? All right. Any questions? Yeah.
I have, if you could remind me again, what exactly is F? What are you trying to do?
All right, so F is sort of you can think of it as an idealized function that ideally we would like to learn it. We are not able to. It's a function that maps a sequence to a 3-dimensional structure of each of them. Right? Instead, we'll be more modest and we'll try to predict contact maps. Right? Okay, so just briefly about secondary structure. So like I said, this is an easier problem and there are many successful programs for that. So what do I mean by secondary structure? It turns out that the protein, locally, most of the proteins are either folded in what's called alpha helix. All right, so these are these things that are turning around. And that's a red thing over there at the top right corner, or what's called beta sheets. Right, these are things, the blue ones over there. And so these are sort of structural motifs that repeat all the time in the protein structure. These are the two main motifs, and there are also other things like protein loops, etc. But that's the main thing that people are talking about when they're doing secondary structure. And then there are programs that you feed them the sequence, and they can predict the secondary structure. That this is how the output looks. So for each place in the protein, we see if it's an alpha helix or a beta sheet, right? And why do they work? Because most of the information is local. So being an alpha helix or beta sheet is usually determined only on the local context, local sequence context. And if you have something far away, it usually does not change the local structure. All right. So this sort of gives us the local structure, the secondary structure, but we also care about the global structure, and this will be given, hopefully, by the contact map. Now, I want to phrase the problem of we are tackling here as a learning problem with auxiliary information. And what do I mean by that? So in standard machine learning problems, we usually have xi, yi in supervised learning, right? So xi are our features, and yi are our labels, and we assume some joint distribution d for xi and yi. Now, the optimal classifier or regressor, if you know d, all right, and for certain loss functions, is simply taking the conditional expectation of y given x, right? That's your optimal f. However, f can be extremely complicated, like, for example, in our case. So it may be hard to learn. But the important thing to notice is that x determines y, right? So if we are in the deterministic case, y is simply f of x. And if we are in a probabilistic case, then if you know x, you don't really need any additional information to know y, right? So mathematically, you can write it as, suppose that someone give me auxiliary information, x augs, right? So the additional variables, then y is conditionally independent on x augs given x, right? And so that's sort of a general theme. For example, in our case, suppose that someone give me the molecule, right, the amino acids, then this sequence determines, again, by the laws of physics, the protein fold. If you'll tell me the sequence of some homolog of this protein, let's say I have the human sequence and you'll tell me what is the mouse sequence, then, you know, physics doesn't care that there is a similar protein in a mouse cell, right? So it will not change the fold of the human molecule, right? So if we knew the joint distribution of x and y, or if we knew f, we would not need this auxiliary information. However, because this function from x to y may be very complicated, and because we don't know this function of the joint distribution, we may use additional information. It could be noisy, and the relation between this information to y, or to some summary statistics of y, or some properties of y could be much noisier, but hopefully it will be simpler to learn, right? So because we don't know the true mapping or the true joint distribution, we are going to use additional information. Even though if we had known them, we would not need that, and we would just need to find a function of x that predicts y, right? So this scenario happens in a few learning problems, although I looked and I couldn't find a theoretical or methodological treatment of this type of learning in machine learning or statistical literature. So I'll be happy to know if anybody knows about such treatment. And I'll just give you another example, not from biology at all, for this scenario. So now in the computer vision community, there is a challenging problem of taking an image and automatically describing it. So the input is an image, and the output is a sentence describing what's in the image. And there's a remarkable progress by the computer vision community. So for example, this picture of a motorcycle, there is a program that after doing learning can output this sentence. You see a person wearing a red and white uniform, is racing a motorcycle, etc., etc. So doing fairly well. Now, how does it do it? So it learns some function from the image, so from the pixels of the image, pixel values of the image, to strings representing text. Now, suppose that you are Google or Facebook or I don't know who, another large company, and you want to really annotate automatically all the pictures on the web using such a system. Well, you can do that. You can learn the fairly complicated mapping from images to sentences, but sometimes you have auxiliary information. For example, suppose that whenever you have a picture, it lies in some file, and you can just look at the file name, right? So many times people who put these pictures on the web will put an informative name, right? So example, motorcyclist, right? So of course, this is a noisy description, right? So sometimes it's useful, sometimes not. But still, the mapping from this description to what we care about, the sentence, is much, much simpler. Why? Because the human already did the work for us in this case, right? So the machine learning system that you're going to build to predict the sentence from the image could benefit greatly from this auxiliary information. Is that clear? All right. So in analogy, in our case, we have the sequence, and we have a fairly complicated function that maps sequence to structure. But we can now add auxiliary information, in this case, multiple sequence alignment. So we basically look at homolog proteins of the query protein in different species, or it could be in the same species. They can be paralogs, right? So we're looking at protein families. And using this information, right, even though, like I said, for all that matters to determine the structure is the first sequence, the query sequence, but still, from using this information, we can get a simpler relation to properties of the protein, and in particular, the contact map. Now, I'll just describe what does the contact map mean. So here we have, if the protein length is D, so we have a D by D binary matrix, and the dark gray entries, they correspond to the value 1. They mean that the two amino acids are contacting each other, and the other one means that they are not contacting each other, right? So this is, of course, a function of the structure. If you give me the structure, I can easily compute from it the contact map. And the relationship between the multiple sequence alignment and the contact map is much simpler than the relationship between the sequence and the structure. Yes?
How do you define contact? Is that like just an angstrom?
Yes, okay, I'll get to that, but basically it's some threshold on the distance.
Okay.
All right. So now we know sort of where we're going. So if we have a query sequence, we need to build a multiple sequence alignment, all right? And then we were going to do some computations on this sequence alignment and try to predict contacts. So I'll briefly describe how our multiple sequence alignments built. This is a topic for a whole new lecture. So basically, we can look at what's called profiles. So profiles are very simple. If you have a gapless alignment, so you have just many sequences of the same length, you can just count the numbers of A, C, D, etc., 20 different amino acids, and you get a matrix of counts. It's called also a position weight matrix or PSSM, right? Many of you, I guess, know it from DNA, but you can do the same from protein. So you get a 4 by D matrix of, a 20 by D matrix of counts. Now, if you have gaps, you can, of course, do also a profile with gaps. So you just have 21 letters, but a more useful and accurate representation of protein is using what's called profile HMM. So there is a model, a hidden Markov model, which has states, and sort of it's a generative model that is used to sort of output a sequence, a protein sequence. And the idea is as follows: you start with the begin state, and then you have a certain probability to output each of the amino acids, but you can also be in an insertion state or in the deletion state, and this sort of states handles the gaps. And there are methods for learning an HMM model from multiple sequence alignment. And in our case, if you start with a query sequence and you want to build a multiple sequence alignment, they're in an iterative process. So you have your query sequence. Yeah, there's a word query missing here. And you go to a large database of sequences, like the UniProt is the standard data set of protein sequences, and you just do search. So you can do BLAST, right? So whenever you have a high similarity, you found a match to the protein. And this sort of gives you the initial family of these are the sequences that are more similar to your query. But now you can take this family, do a multiple sequence alignment, and you build what's called a profile HMM, which is sort of now representing not your query sequence, but the entire family of sequences. And now you can go back to the database and search for other sequences that look like your family, because sometimes it turns out that BLAST search missed them due to all kinds of reasons. And you can iterate the process and by that enlarge your family. So you get more and more sequences that will not, whose similarity to your query sequence was not easily detected on the sequence level. All right, so this is a whole field of how to generate good alignments for a query sequence. And once we have the alignment, we'll feed it into the next stage of predicting contact maps. But which means that it is important to get good alignment. So if you improve your method or either your database or your method for generating alignment, you can get better alignments, and you can also improve on your contact map prediction. All right, so that's the first stage. And there's a whole set of tools based on HMMs for doing all this stuff. It's called HMMer. All right. All right, so now suppose we have alignment. Now we need to define contacts. And these are defined simply as a thresholding the distance. Now, when you look at two amino acids, what do you mean by the distance? You can look at the closest distance or the furthest distance, etc. And there are standard ways that people define distance. So there's what's called the C alpha atom, which is a particular atom in each amino acid. So you can define the distance between the C alpha atoms of two amino acids and threshold it. And in the literature, people are using anything from 6 or maybe 5 to 10 or 12. And I'll show you, hopefully at the end, something about these distances. And you can also use a C beta atom, which is a different atom, etc. Now, how do you evaluate performance? So you can just treat contact prediction as a binary supervised learning problem. So for each pair I and j, either they have contact or not, right? So you have the 0-1 classification loss. But it turns out that what is most informative are the top contacts, right? So if you have D choose 2 possible contacts in a protein, most of them are not present, right? So the number of actual contacts grows only linearly with sequence length. So this means that usually what people in the field are looking at are the top D or D over 5 or D over 10 contacts out of the D square over 2, right? So a computational method should take all the pairs ij, give a score to each one, rank all the D choose 2 pairs by this score, and we are looking only at the top of the list of the ranking, let's say at the top D, and to evaluate performance, we are comparing it to the true contact map and measuring how many of the top D that we reported are actual contacts in the structure. All right, so that's a sort of a direct evaluation. There's also an indirect evaluation, and some people care more about that, which is, okay, you gave me a contact map, now I can feed it into a 3D structure prediction program as constraints, and these are very powerful constraints, because if you have your chain, your protein, and you force this amino acid to touch this amino acid, it reduces your degrees of freedom, and then you ask, well, how much did my 3D structure improve due to the contacts that this method gave me? All right, so I'll describe now, I see that I don't have much time. I'll describe now 2 methods, unsupervised methods that use a multiple sequence alignment to predict contact maps. Now, the idea is sort of obvious and it's not new, which is if we have 2 amino acids that touch each other and one of them mutated in evolution, then the other one also has to mutate so they match, right? So if once one of them acquired a mutation, maybe the local biochemical properties of the 2 don't match and the contact will be ruined. So we need a compensatory mutation in the other one. So if we look at the multiple sequence alignment, we can look at the 2 columns and start and look for co-evolving columns. So we need some score for co-evolution. So like I said, this idea is fairly old, but it really became powerful about less than 10 years ago. And there were a few methodological improvements. The main one was to distinguish between direct and indirect effects. So if we have A is contacting B and B is contacting C, then we expect to see correlated evolution between A and B and also between B and C. But then we'll also see a correlated evolution between A and C. So if we calculate some score measuring the correlated evolution of A and C, we may conclude that A and C are in contact, but this conclusion will be wrong, right? So how do we distinguish between direct and indirect contacts? Well, it turns out that the language of graphical models is helping us here. In particular, if you'd assume for a moment that we had Gaussian random variables, so for a Gaussian vector, if Z is a Gaussian vector with covariance matrix sigma, then what matters for this direct or indirect interaction is not the covariance matrix, but the inverse covariance matrix, also called the precision matrix, theta. And 2 coordinates, Zi and Zj, are independent, conditionally independent, condition on the other variables, if and only if the entry theta ij in the precision matrix is 0, right? So what we want to do is fit a covariance matrix such that if we want the contact maps to be sparse, right, because we assume that most pairs are not in contact, we would like to fit a covariance matrix such that the inverse covariance matrix, the precision matrix, will be sparse. Now, how do we do that? There is an algorithm to do that from 2008. It's called the graphical lasso. All right, so I guess many of you know about lasso for regression. And in lasso, we are trying to fit some coefficients, beta, and we add a penalty that is L1 penalty. So this lambda times theta L1 norm. And what this penalty is doing is it's sparsifying our solution. So it's causing many of the coefficients to be 0, right? So graphical lasso is a utilization of the same idea for fitting sparse inverse covariance matrices. So we have this like a lasso penalty for the entries of the precision matrix. And right, so here, so it's the sum over all the entries of the precision matrix. And this gives us some penalized likelihood function that we optimize. All right. And there are some other technical details, like shrinkage of the sample covariance. I won't go into that. And that's basically what the Psychov method is doing. It's taking the data, the multiple sequence alignment data, and is fitting a sparse inverse covariance matrix. Now, how do we rank the pairs? So for each two positions, I and j, we need to sum. Oh, you can't see the mouse. Okay. So, I mean, the matrix is built of all the columns I and j, and also all the amino acid identities A and B. Each one of them have 20 possibilities. Now, if you care about two columns, you need to sum over the identities of all the amino acids, and you get this PCIJ score. You normalize them because you can have one column which has many, many high scores or many, many low scores. So you normalize by the average of the row and of the column. And this gives you the score PCIJ. These are the scores that are used to rank all the D choose 2 possible candidate contacts. And you just take the top, and these are your prediction. Now, if you look at the performance, then this is from their paper. So on the x-axis, we have the performance of just doing mutual information between two columns, which is not taking into account these direct versus indirect effects. And on the y-axis is their performance, and as you can see, it's clearly much higher for almost all the protein. All right. So that was Psychov from 2012. Now there is a similar method called direct coupling analysis that was published roughly in the same time, 2011. Due to time, I will not describe it, right? So, but I mean, I'll tell you just the main difference. Psychov is sort of taking its motivation from Gaussian random variables, or even though in our problems the random variables are not Gaussian, they're binary, and direct coupling analysis is using a discrete graphical model, not Gaussian. So there are many different details about this method, but the principle is the same. It's trying to fit a sparse graphical model, right? So I'll skip all the details of the direct coupling analysis method, just give you the reference. It's Morkos et al. From 2011. Right. And the computational complexity, that's important. It's more computationally heavy than Psychov, but still fairly scalable, so it's proportional to the number of sequences cubed. All right, in particular, the output of this method, it's called direct information, which is, you can think of it as some modification of mutual information, but when you condition on all the other variables. Right? And you rank your pairs by the direct information rather than the mutual information. And they also show a great improvement. So this is the direct information results, and these are the mutual information results. And red are correct contacts, and green are incorrect contacts. And you can see they improve quite a lot versus mutual information. All right. Okay. So how am I doing on time? 5 minutes? 3. All right. All right. So these two methods were very nice, and there are a couple other ones using similar ideas. Notice that all these methods are unsupervised, right? They are trying to fit some graphical model to the alignment. They are not really using the true contacts, the true structures. In the recent few years, there have been improvements over this method that use basically two ideas. One is supervision, so we're going to use known structures and known contact maps to fit our models, to predict them. And the other one is using neural nets. So one is called Metapsycho, which is by the same authors of Psychos. What they basically did is, and this is sort of common to all of these methods, is you run the simple methods first. So for example, you run Psycho of DCA, you can run also a secondary structure prediction program, and you use your profile. All this sort of simpler program give you features, new features, and there are single amino acid features like a profile, so it's a frequency of each amino acid in this location, and there are also pairwise features, like for example the prediction of direct coupling analysis for a particular pair. Now you take all these features and you throw them in some way, in some clever way, into a neural net, and you try to predict the contacts, 0, 1 contact, either a contact or not. And as you can see, you get an improvement. I mean, on the right side, you see that the highest bar is Metapsycho, and the lower ones are the sort of simpler method of Psycho or direct coupling analysis, etc. Right? So by throwing in different features, you can improve your results. There is a even more recent method which improved the result further by Wang et al. From Jinbok Su's lab. And they use a similar idea, but they use the deep network, I think, for the first time for this problem. And importantly, they also use convolutional networks, both for the single feature, the single amino acid features, and 2D convolution for the pairwise features. Now, you can think of these convolutions as one of the things they are doing is averaging information locally. Because if you know that there is a contact between, let's say, amino acid number 7 and amino acid number 22, then, you know, what does it mean? It means that they are close in 3D structure. It means that it's more likely to have a contact also between amino acid number 8 and number 23, right? So it's local. So this convolution layer is pulling information and is sort of once you have good evidence for contact here, it also improves your posterior, if you want your posterior, or the likelihood that you'll predict contact over here. Right. And you can see also the improvements here. So this is the last line is their method. And you can see the precision at different values of L. Now, okay, I'll conclude with a fairly recent development, which was an even further improvement, which happened at the end of 2018. And this was from the DeepMind group at Google. And many of you know them from AlphaZero, right? So it's a program that plays Go and beats everybody in the world. So it seems that they are now doing that also for this problem, right? So there is for those of you who don't know, there is a competition called CASP every year, where there is a ground truth, which is a structure experimentally solved, and then computational groups compete, and they submit their solution and trying to predict the structure. And this year, DeepMind AlphaFold won the competition, and not only they won, they improved quite substantially the accuracy compared to previous years, so there was a big jump in performance. And this is sort of their pipeline. They have a neural net to predict the distance. There's also another aspect I didn't talk about predicting angles between change. And then there is a second layer that uses this information and and predicts the structure. One idea they use, which was also used by Jinbok Su's group in 2018, is we are not trying to predict only simply binary contact, 0, 1s, but we can sort of throw away this somewhat arbitrary thresholding and try to predict the actual distance between two amino acids. And that's why we got these nice colorful pictures instead of 0, 1 pictures. So instead of predicting contact or not contact, we are actually predicting the exact distance between two amino acids. And it's a regression problem for each contact instead of a classification problem. And now for each two pairs, we have a range of possible distances that they must satisfy. And this helps at this next step of predicting the 3D structure. All right, so you can think of this remarkable progress as either encouraging or maybe if you're not in a Google, discouraging, right? So luckily for me, I'm new to the field, but there are academic groups whose entire profession was to improve on structure prediction, and then came this machine learning group from the industry and sort of beat everybody. So the question is, does it make sense to continue and try to compete with them? That's a good question, and I recommend reading, there's a nice blog by Mohammed Al-Karishi, who is one of the researchers in this field, and he talks quite frankly about his feeling and where does he think this field is going. I'll tell you where we're trying to go. So I think if you really trying to predict, trying to compete with large groups who have great people and B, access to massive computing and other resources, it's going to be hard to compete. So instead, you can try to solve, you can still work on this field. I mean, this field is very rich and interesting, but maybe change the problem. So here are a few other problems that one can work on. So you can look not only on contacts within one molecule, but try to do protein-protein interactions. So when two proteins interact, you want to predict where are the contacts, right? You can study structural heterogeneity, right? So if you have a molecule, a protein, so far we assume that it always has the same structure, and but in reality, some proteins have some flexibility, and this is not accounted for by the computational methods. And the last thing, that's something we are working on, and if you are interested, please talk to my student, Omer Ronen, who is here. I don't see him, but he's here. All right, so we're trying to sort of solve this problem, but for the case where the structure itself changes. So if you have a multiple sequence alignment, the assumption of all these methods is that they all share the same structure, and if you have a contact, it will induce coevolution. But sometimes the contact itself changes during evolution, and it appears in some of the alignment and doesn't appear in the others. And we're trying to model and solve this problem. All right, that's it, and sorry for taking too long. And thanks, Omer, and I also want to thank other people who helped me learn this field and prepare. So Yair Heller, Jerome Tubiana, and Dina Schneidman at the Hebrew University. And I'll be happy to take questions.